So this visual interpretation is an FDA-approved method to assess tau PET images, right? So clinicians need to apply this visual interpretation method in order to classify a tau PET scan as positive or negative. On the other hand, in research, we don't do this typically, right? We don't assess the scans visually. We applied, fully automated methods in which we quantify the so-called SUVR in certain regions of the brain. And the idea of this study was to see, okay, so are these two methods comparable or not, right? And what we found is that they are not comparable, okay, in particular in the cognitively or in the unimpaired stage, right? So, there were lots of disagreement between the visual interpretation and quantification in cognitively normal individuals, basically because the patterns of tau deposition were pretty heterogeneous, right? You could find like focal deposition only in the occipital cortex, only in the parietal cortex, and the traditional quantification methods will miss all these cases because they focus mainly in the temporal lobe, right? So that was the main conclusion of our study, is that visual interpretation, okay, the FDA approved method is not comparable in general. Okay, it's more comparable in impaired individuals because, when you go to later stages of the disease, tau is basically everywhere in the brain, and therefore it's more likely that both approaches actually agree, right? But in general, these visual interpretation and standard quantification techniques are not comparable. Okay, so the message here is that, they're not comparable, and we need a better quantification approach that basically is able to quantify the amount of tau tangles in the brain, but also have better agreement with visual interpretation and the fact that with visual methods we can analyze the entire brain, right? Not only the temporal lobe, which is what these quantification approaches actually do.